The plasma membrane is selectively permeable. It lets some substances through and not others. The hydrophobic interior of the bilayer is one reason membranes are selectively permeable. Hydrophobic molecules, those that are soluble in lipids, can easily pass through the membrane. In addition, small molecules like O2 can sneak between the phospholipids of the membrane. On the other hand, large hydrophilic molecules like glucose and ions such as sodium ions and hydrogen ions cannot pass through the membrane unaided.
So as you can see, the plasma membrane is a semi-permeable barrier made up of phospholipids such that there is a hydrophobic core and hydrophilic surfaces, right? There's a hydrophilic surface facing the extracellular fluid and one that faces the cytoplasm. And then embedded within this phospholipid bilayer are proteins which act as pumps, channels, gates, and receptors. And what they do is they control the movement of stuff in and out of the cell, and they tell the cell about what's going on outside, right? And they also allow the cell to respond to events outside the cell in such a way that the cell is more likely to survive, right? In addition to these proteins, the integral proteins, which pass all the way through the plasma membrane, and the peripheral proteins, which are on either the cytoplasmic or the extracellular side, we have carbohydrates that are attached to a lot of the phospholipids and cholesterol embedded in the membrane. The cholesterol serves as a way to control what parts of the membrane move around the most, so it acts sort of like a doorstop. And the carbohydrates are basically a chemical ID tag that indicate whether or not that cell belongs in the organism, and if it doesn't, those chemical ID tags will serve as a clue for the immune system to move in and destroy those cells. So, for instance, when our cells get infected, one of the things that can happen is that the cell surface can change in such a way that the immune system moves in and destroys it before it gets a chance to generate more damage. Okay? Membrane lipids are 75% phospholipids, where the heads are polar and hydrophilic, and the tails, which are nonpolar and hydrophobic. And then 5% glycolipids, which are lipids with polar sugar groups on the outer membrane surface, and 20% cholesterol, which, as we already stated, is important for controlling how much the membrane moves around, right? There are, for instance, some integral proteins and peripheral proteins that work best as dimers or trimers or tetramers, and it's important that if that's how they work best, that if they're embedded in that part of the membrane, that part of the membrane doesn't move around very much. So sometimes we call those lipid rafts that are floating on the sea of phospholipids. Okay? And it's all, again, to help out with their function, right? The membrane proteins, as we stated, allow communication with the environment. They're about half the mass of the plasma membrane. Their specialized membrane functions help the cell adapt to its changing internal and external environment. Some of them float freely, and some of them are anchored to the cytoskeleton, and some of them are in areas of the phospholipid membrane that don't move much because there are a lot of cholesterol inserted in that region. Okay? And of the two types that we noted, the integral proteins go all the way through the plasma membrane, and the peripherals are on either edge. Okay? So let's take a look at membrane proteins.
Transport proteins carry molecules from place to place. The example shown here allows certain solute molecules to enter the cell. Hemoglobin is the transport protein that carries oxygen in the blood. Receptor molecules bind to signal molecules and can then emit second messengers which trigger changes inside a cell. Receptors are thus important links in the system of communication among cells. Some signal molecules, such as hormones, are also proteins. Structural proteins have many functions. Like tent poles and ropes, They shape cells and anchor cell parts. They may serve as tracks along which cell parts can move. They bind cells together, making organized units such as muscles, ligaments, and the tendons that bind muscles to bones. The silk of spiders and the hair of mammals are also structural proteins.
So the 6 functions of membrane proteins include transport, serving as receptors for signal transduction. That's basically like getting the chemical news outside and then relaying that information to the inside of the cell so it can react to it. Attachment to cytoskeletal and extracellular matrix components, enzyme activity, intercellular connections, and cell-cell recognition. Okay? An example, right? Attachment to the cytoskeleton and the ECM, extracellular matrix. So the cytoskeleton can attach to these proteins that are embedded in the cell membrane, and that can serve to anchor the membrane to its neighbors or to what we call the basal lamina in some cases, which is a non-cellular secretion that acts kind of like glue. And that maintains the cell shape and it sort of fixes its position. And others are critical in the movement or the binding of cells to each other. Okay, so let's take a look at that.
An enzyme is a protein that changes the rate of a chemical reaction without itself being changed into a different molecule in the process. Enzymes promote and regulate virtually all chemical reactions in cells.
Yeah. In a pinch, you can use a piece of cardboard or the back of a book. Yeah.
You just hanging out.
Making a podcast.
How late are you yourself?
Oh, not too late.
Diffusion is the tendency for particles of any kind to spread out from where they are more concentrated to where they are less concentrated. This process can also be described as molecules moving down their concentration gradient. Diffusion across a biological membrane is called passive transport, since the cell expends no energy to move the molecules. Oxygen and carbon dioxide molecules move into and out of cells by passive transport.
So another function, right, intercellular joining, is one of the ways that we establish tissue barriers and allow tissues to function as a group, right? Some membrane proteins called CAMs provide temporary binding sites that can guide cell migration, and others are more permanent and generate junctions that can be either semi-permeable or impermeable. Some glycoproteins serve as ID tags that indicate whether or not that cell belongs in the body and is recognized and tolerated by the immune system. So that's sort of like, again, a chemical ID tag for the cell, telling it that it belongs. We talked about the lipid rafts, right? They are basically areas where there's less movement of the phospholipids than in other areas. They usually have more cholesterol around. You may see sphingolipids as well as phospholipid. And they function as platforms for membrane proteins that have to work closely together in order to function, or for areas of the cell that can undergo invagination, such as in vesicle transport. Okay? The glycocalyx is just a sugar coating on the cell surface that, again, can serve as a recognition molecule. Okay? This allows, again, the immune system to determine whether cells belong to the body or not. These recognition molecules can change in the event that the cell becomes a cancer cell, or sometimes change in the event that it gets infected by a virus or a bacteria that tries to do its dirty work inside the cell. Okay? Cell junctions are just attachments between cells so that they can work as tissues. And some cell junctions are non-existent, right? Meaning that there's matrix between the cells and the cells move around. Those would be liquid connective tissues such as blood and lymph, and then the only motile cell in the body, the sperm cell, and the egg cell after it's been ovulated from the ovary. Okay, but a lot of them are held together by physical junctions that include tight junctions, desmosomes, hemidesmosomes, and gap junctions. Okay, so when we look at tight junctions, we're talking about integral proteins that fuse and form an impermeable barrier so that nothing can move between the cells, and the only stuff that's going to pass is going to be stuff that actually goes through the cell as a result of properties of the apical and the basement membrane. Okay, so you might find tight junctions in places like the epithelium of the skin. Okay, you might find it lining the urinary bladder. Okay, all areas where we want to keep the stuff inside, and the stuff outside. All right, other than what we want to permit to go through the cells. Desmosomes are like spot welds and anchor the cells together at structures called plaques, which are attached to cytoskeletal elements. All right, and essentially what happens is that filaments such as keratin can run through the cytosol to the opposite plaque and provide sort of like a fence going through the edges and the center of each cell, and that makes it physically tougher, less likely to tear, less likely to end up with damage. All right, and again, you might find these in epithelial linings. All right, basically cells that line organs or spaces in the body where we want to keep the material inside, and we don't want it to leak into other areas. Gap junctions are different. Gap junctions are communication junctions. They're like tunnels between adjacent cells, and they allow the movement of charged particles, ions, from one cell to the other, and other small molecules, and even perhaps messenger RNA. And so these are communication networks. We find these in places like smooth muscle, and we also find it in cardiac muscle, and occasionally we will find it in certain nerve networks. Okay, these are electrical junctions, and they allow very rapid movement of electrical signals from one cell to the next. That's their primary purpose. Okay? The plasma membrane is the barrier, the outer barrier of the cell, and outside the plasma membrane is the matrix, which can either be a fluid, a solid, a semi-solid, okay, depending on the tissue type. There's lots of stuff in the interstitial fluid, nutrients, building blocks, and waste products, as well as electrolytes. And the plasma membrane lets the cell take what it wants from the outside and pitch what it doesn't want from the inside, and as a result continue to function as a living unit. Membrane transport is the way that this is done, okay? And membrane transport is essentially a method of moving the material from one location to another so that the cell can either get what it needs or throw away what it doesn't want. Two ways that substances can pass across these membranes include active and passive transport. In passive transport, the movement is always down the concentration gradient from most to least concentrated, and in active transport, it's always in the opposite direction, and as a result, it requires energy, which can come either from ATP directly, or it can come indirectly from a gradient set up by a primary transport that uses ATP. A molecule will passively diffuse through a membrane if it's lipid soluble, or if it's extremely small, or both, or if it has a carrier, a special tunnel, usually made out of protein, that's going to let it through. The two types of passive transport include diffusion and filtration. In diffusion, a solute moves from a more to a less concentrated region down its gradient. It can either go through the membrane if it's a lipid-soluble compound, or it can go through a tunnel if it is a charged or a polar compound. Okay, it is a passive type of movement as it goes from high to low concentration. Filtration is the result of hydrostatic pressure against a surface, and that's what drives material through openings from one location to another. And we see this in places like the kidney. Okay? In diffusion, what's happening is that solutes are distributing themselves such that there's no longer a concentration gradient for the solute in areas of a fluid that are connected to each other or across a membrane through which they can move. All right? The speed is influenced by the size of the molecule and the temperature. All right? And so let's look at a little bit about diffusion.
The plasma membrane is permeable to water molecules, and the movement of water into and out of cells is critical to life. Diffusion of water molecules across a selectively permeable membrane is a special kind of passive transport called osmosis. Plant cells are surrounded by rigid cell walls. When plant cells are exposed to hypotonic environments, water rushes into the cell, and the cell swells, but is kept from breaking by the rigid wall layer. The pressure of the cell pushing against the wall makes the cell turgid and is the desired state for most plant tissues. For instance, placing a wilted celery stalk or lettuce leaf in a hypotonic environment of pure water will often revive the leaf by inducing turgor in the plant cells. Animal cells lack rigid cell walls. When they are exposed to hypotonic environments, water rushes into the cell and the cell swells. Eventually, if water is not removed from the cell, the pressure will exceed the tensile strength of the cell and it will burst open, or lyse. Many single-celled protists living in freshwater environments have contractile vacuoles that pump water back out of the cell in order to maintain osmotic equilibrium and avoid lysis. Plant cells are surrounded by rigid cell walls. When plant cells are exposed to hypertonic environments, water rushes out of the cell, and the cell shrinks away from the rigid wall. These cells are dehydrated and lose most or all physiological functions while in the shriveled state. If cells are returned to isotonic or hypotonic environments, water re-enters the cell, and normal functioning may be restored. Animal cells lack rigid cell walls. When they are exposed to hypertonic environments, water rushes out of the cell, and the cell shrinks. The resulting cells are dehydrated and lose most or all physiological functions while in the shriveled state. If the cells are returned to isotonic or hypotonic environments, water re-enters the cell, and normal functioning may be restored.
Now, certain polar molecules are transported passively by binding to protein carriers that are embedded in the membrane. That's the only way they'd be able to get across because of the high energy barrier necessary for them to get through the hydrophobic core of the membrane. So they need a helper, right? And the protein channel is going to be the helper. So they move through these water-filled channels, which are integral proteins that carry them from one end of the membrane to the other, down their concentration gradient. They're specific for their substrate, and they can be saturated, meaning you can get to a maximum transport rate as a result of filling all of the transport membrane proteins, and then we can't get the material to go in any quicker than that, right? And that's called saturation when they're all engaged. Okay? Now, these channels, right, can either be open all the time, in which case they're called leak channels, or they can be controlled by a physical change at the membrane, and in that case they're called gated channels. Okay, so channels can be gated by voltage changes, they can be gated by mechanical changes, and they can be gated by ligand binding changes. Okay, so those are all ways that these gated channels can open or close. All right. The movement of a solvent across a selectively permeable membrane, and the solvent in living systems is always going to be water, right, is called osmosis. So osmosis is just a fancy term for the diffusion of water from where it's most to least concentrated. So water is always going to diffuse from where there's more of it to where there's less of it. Another way to think of this kind of movement is that water chases solutes, okay? So the way that the water gets across, there's two ways. It can go through the lipid bilayer because it's small, but it also can go through a water channel called an aquaporin, okay? And what will always happen is that the water will attempt to equalize its concentration on either side of the membrane as a result of, again, flowing down its concentration gradient, okay? So the more solute there is in the solution, the less water there's going to be, okay? And so the highest water concentration is going to be pure water. Anything dissolved in pure water is going to mean that there's fewer water molecules per volume. And so if you have a solution on one side of a barrier and pure water on the other side of the barrier, and the water can move across the barrier and the solute can't, what's going to happen is that the pure water is going to move across that barrier until the concentration of water molecules on either side is the same, or until so much pressure builds up on the other side that the hydrostatic pressure is able to counterbalance the osmotic pressure. Okay, so what you would see in that situation is one side of this chamber rising as the water moves into it from the pure to the solution side, and the other side of the chamber, the fluid level would go down. Okay, so that's, again, a passive process, and that is how water moves around in living systems, including in the human body, right? Basically, where solutes go, the water will follow, and the main solute that guides where water goes is sodium, okay? So where the sodium goes in our body, the water will follow it, all right? So let's take a look at osmosis.
The image on the left is human erythrocytes, or red blood cells. The image on the right is enhanced with filters. These living cells are in isotonic solution. Note the lozenge shape. Now the solution has been changed to have a high salt concentration. It is hypertonic. The red blood cells shrivel and become crenated. Finally, the solution has been changed to plain water. The cells swell and burst, leaving empty membranes behind. This phenomenon is called hemolysis.
Now, tonicity just refers to the solute concentration relative to a point of reference, right? And the point of reference is going to be the inside of the cell in this case. So in a hypotonic solution, the water concentration outside the cell is greater than inside, and the cell will tend to swell up and rupture. In a hypertonic solution, the water concentration is greater inside than outside the cell. The water will tend to leave and the cell will shrivel up, and that's called crenation, C-R-E-N-A-T-I-O-N. And then in an isotonic solution, the rate of water movement into and out of the cell is the same, and the cell shape will be retained, okay? So this is why, for instance, when you get an IV, we never give you distilled water to replenish fluid, because if we did, we would rupture your red blood cells, they would release their hemoglobin, which would jam your capillary beds, cut off blood to downstream tissues, and you would die, right? So that would be a huge mistake. We always give you something called isotonic saline in most cases, right, which has the same water concentration as your plasma, and as a result won't pop your red blood cells or won't shrink them. And the body always goes through great pains to try and maintain an isotonic situation around cells and tissues whenever it can. And so the body systems that work to do that include the urinary and the integumentary system, okay? And what they do is they try to maintain fluid and electrolyte balance so that we don't create conditions that will cause the cells and tissues to either rupture or to shrivel up and die, right? So this is another one of those, what we call homeostatic variables, right? We have to keep the water and electrolyte concentration within limits compatible with life, or else it's going to be lights out for you.